Hi everyone, my name is Julia Ribeiro. I'm a student at FIU, and I'm here today to talk about my review paper on the effects on pulmonary and systemic circulation of microplastics on the human body. So when plastic materials break down, microplastic and nanoplastic particles are actually small enough to enter animals, overflow the environment, and even the human body. The particles are so small that they could be either inhaled, ingested, or even absorbed by the skin through contact in a process similar to endocytosis. So these particles, they can actually originate from so many different sources of larger plastic materials. They can be present in cosmetics, textiles, packaging, industrial waste, and the increased exposure of these particles in the environment, plus their ability to enter the human body and deposit in different tissues, is now raising concerns about the potential health risks. Research on the potential health risks caused by microplastic accumulation actually suggests that they can induce oxidative stress, inflammation, and cellular toxicity, which can lead to so many diseases. The microplastics are small. Once they get inhaled, they get into our bloodstreams and then into the cells, causing cell damage. Due to their versatility, affordability, and easy manufacturing, plastics are present on most of the items we use daily. And once these items break down, microplastic and nanoplastic particles are actually exposed everywhere. So research on ingested microplastics have said that microplastics are constantly transferred from one trophic level to the next, and that by 2050, nearly every seabird will be consuming plastics. Now, research on inhaled microplastic says that once they enter the pulmonary system, they can either accumulate within the lung lobes in different concentrations and regions, or they could be translocated to other tissues and cells throughout the bloodstream in systemic circulation. So this is a diagram explaining a little bit the exposure and translocation of microplastics. Over here we have some of the different sources, and once they are inhaled, we can see that they can come in contact with the bloodstream and the lung tissue and be carried out to the rest of the body with human hemoglobin. If microplastics are ingested, they can also translocate through the body by transferring from one tissue to another by translocating epithelial tissues. So over here we can see they go through the gut tissue into the bloodstream. And if microplastics enter the human body through endocytosis or absorption in dermal contact, a similar process also happens where they can cross membranes and get into the bloodstream, where they'll be carried out to the rest of the body or either accumulate in different tissues. Experts are now emphasizing the prevalence of microplastic pollution on a global scale and how it's making human exposure inevitable. So this study was actually done to obtain flow patterns in order to assess associated potential health risks that comes from microplastic accumulation on lung regions. Some of their findings was that inhalation intensity of microplastic particles directly controls their deposition rate. Over here we can see with a total of 3.80%, 5.10, and 5.81% of deposition rates and inhalation intensities of 7.5, 15, and 30 liters per minute. So as inhalation intensity increases, so does the deposition rates. It was also shown that different shapes and sizes of microparticles actually control where they are deposited. So larger particles have a higher deposition rate, as we can see over here. In this graph, A represents the smallest particles of 1.6 micrometers and the larger particles of up to 5.56. So we can see as the size of the particles increases, so does their deposition rates, starting very little over here and increasing as the size gets bigger. We also see that as respiration rate or inhalation intensity increases, so does deposition. So at lower flow rates of 7.5, particles have enough reaction time to align and avoid collisions, which allows them to enter and deposit specifically on the lower lobes over here. and avoiding the upper. While at quicker or higher intensity, we can see that they don't have enough time to react and the particles end up colliding with the walls and entering bifurcation. So they accumulate specifically on the upper lobes of the lungs. The trachea is also shown over here, and it has a less complex structure when compared to the rest of the lungs. And we can see that this has a very stable and smooth airflow without really big disturbances, which allows the particles to move past it very stable and very smoothly and not accumulate as much as they do on other lung regions. So we can see that accumulation on the trachea is only really seen at the first bifurcation over here, not as much compared to other lung regions. Now, this other study showed that nanoplastic particles were actually found to disturb gene expression, to activate inflammatory pathways, increase toxicity, and decrease viability of cells. They also induced apoptosis and cell cycle arrest, especially in the S phase. So this study used two different sizes of microplastics, specifically polystyrene in a 25 nanometers diameter and 70 nanometers diameter. So with the size of 25, concentrations above 20, and in the size of 70, concentrations above 100, actually showed a significant toxic effect on human lung cells, specifically A549 cells. And this showed in a dose-dependent manner. So as the dose increased, so did toxicity. As incubation time increased, it was also shown to increase the effect of these nanoplastics on the cells, and they became even more evident. We can see over here this happens for both apoptosis and cell cycle arrest, specifically in the S phase, in different incubation times of 2, 4, and 8 hours. Lastly, nanoplastics were also shown to disrupt gene expression. They actually increased upregulation of genes that are involved in proliferation pathways like CCND and Ki67, as well as genes for nuclear transcription factors, shown over here at the end of the table. Microplastic particles have actually been found in very different cells and tissues of the human body, including the placenta and milk samples, which suggests that these particles can translocate through the body to accumulate in different regions. This research was able to confirm that this translocation does occur and that there is a binding interaction between the most common microplastic particle, PVC, and bovine hemoglobin. This is a very important protein present in the bloodstream of pigs that shares 90% of the same amino acid sequence as human hemoglobin. So this suggests that this binding interaction between bovine hemoglobin and microplastics can actually happen in humans within human hemoglobin, which suggests a possible mechanism of translocation. This research confirmed that the binding interaction occurs. Over here we can see in this TEM image showing the complex formation, and it also shows that this binding interaction is indeed spontaneous. So it's shown over here by the negative delta G, negative delta H, and positive delta S in the thermodynamic parameters, showing that electrostatic forces play a role in the complex formation. Now, it was also shown in figure A over here, 2A, that the PVC microplastic, as the concentration increases, can actually induce structural changes in the bovine hemoglobin. This is shown by the reducing alpha helical content in the protein, which can change the protein's secondary structure and even affect its function, which can prevent it from binding to other very important molecules like oxygen. Other than translocation, this study actually researched myocardial damage in chickens specifically, and they found that the polystyrene microplastic particles actually promoted myocardial inflammatory injury in chickens. So low, medium, and high concentration of microplastics had a more obvious abnormal morphology in cardiomyocytes with higher concentration. Over here we can see that this morphology was due to many different inflammatory cell infiltrations and myofilament arrangement, which are shown by the different arrows. Over here, you can see the green boxes, and they increase with the higher concentration. Similarly, the same concentrations were also caused to increase the mitochondrial damage and apoptosis, which are shown by the colorful arrows over here that increase with the higher concentrations, specifically when compared to the control group that has no arrows. Now, myocardial injury area was actually confirmed and correlated to the rest of the experiment that showed that the higher concentration group did in fact have a higher myocardial injury area, showing that this damage does correlate to higher damage in the heart. The same study found that increased concentration of reactive oxygen species were found in primary cardiocytes with higher concentration of microplastic, and they can be seen on this graph and on the image over here, as well as that cell viability of cardiomyocytes ended up decreasing with both time and concentration of microplastic. So as the concentration increased and time increased, the viability of these cardiomyocyte cells decreased, showing that there has indeed toxic effects of microplastic accumulation on heart tissue. In conclusion, the reviewed studies highlighted the significant effects of microplastic inhalation in lungs and heart tissues. The studies showed that inhalation intensity, shape, and size of microplastics directly controls the deposition rates and their locations within the respiratory system, and that this accumulation was linked to increased RLS formation that led to many disturbances in gene expression, upregulating inflammatory and apoptotic pathways over here, including DNA damage, leading to cell apoptosis and cell cycle arrest. This review showed that microplastics are small enough to either accumulate on the lungs or be transferred to human hemoglobin and translocate throughout the body. The results emphasize that negative consequences of oxidative stress due to the microplastic accumulation has a potential long-term health impact on lung tissues and heart tissues. Studying this process of translocation and accumulation throughout the human body and the mechanisms of organelle disruption can lead to a better understanding of these disruptions and possible long-term effects for humans. This review is essential in guiding future research to understand implications of these biological disruptions, as well as help in developing tools and tests for microplastic detection and exposure. Thank you for listening. Any questions?